Use Daily Mail.
Yeah.
Okay, so Gustav is going to talk about this project now.
Right, this is up and running.
So,
Did you?
I don't know.
Should I wait or?
I don't know.
Is it running?
Looks like it.
Yeah. Okay. Good. All right. So, weather forecasting, sort of. Right. So, some background on what we're doing. So, this group is me, Gustav, and Dennis and Mahmoud, which are not here. They are on vacation right now. And this is their research area. I'm just the guy from IT. So I'll do my best of sort of presenting what we're doing.
Sometimes it's very good to have the research presented by someone who doesn't know
much
more about it.
So I can give you now some hopefully good background to explain this. So basically, as you know, renewable energy is the latest big thing, right? You have solar power and wind power, and these are great. But one of the big drawbacks of them are that they are unreliable. Basically, if you have a cloudy day, like right now, you get less solar power, or if there's no wind, you get less wind power. And this is a bit of a problem, because if you think about the power grid, it basically has two limits, right? You have some capacity in the power grid, and then you have some demand. And the power level of the grid always needs to be in between that. Ideally at the demand, but right above it. And the capacity of the network is, of course, fixed. There's only so much power it can take before things start blowing up. But then the demand will change throughout the day and throughout the seasons. So now the problem is that if we only had solar power or solar panels everywhere, then when it's cloudy outside, then we may go below the threshold of the demand, and then things stop working. So what you need to do is basically you need some backup power system. Energy systems that's like nuclear or biofuel or whatever that you can sort of fill the gap with. Okay, but the problem is that you don't know. A week from now, how much solar power will we be generating, or how much wind power will we be generating? But it takes roughly, I think it was like a week to power up a nuclear power plant. So there's some problem there. You want to know how much power will we predict in the future, or produce in the future, so that we can start up the nuclear power plants to sort of fill the gap if necessary. So this project this project has been about that. So Dennis is collaborating with some, researchers who have in Engelska Parken, sort of a solar panel with the tiny wind station on it that measures like the wind speed, temperature, stuff like that, and also measures the produced power by the solar panel. So that thing is generating data every 5 seconds, and that's the data we've been working with. And the goal of this project has been to, given that data, try to predict what is the power generated in the future. We don't have any specific targets on like how far in the future or anything like that. We've just been playing around with horizons. So I'll just walk you through basically what we've done now. So stuff, I realize now there's a lot of stuff missing here. Like I don't think I have any graphs visualizing the data, so it will be a bit boring to look at. And also I have a pre-processing step before this notebook, which is just a bash script I set up, which takes all the data comes in JSON files, and I think we have for like 5 months basically, and it's 4 gigabytes of data, and it's too big to upload to the community edition. Where it turned out like we only need a couple of fields in the JSON file to do our prediction. So by us removing junk, we could reduce the size by 96%. So there's that. So once the data is clean, then it's quite straightforward. We load the data. We have our these are the different fields we are extracting. As you see, they're very descriptive. Right, and then we do some pre-processing. So basically, we get data every 5 seconds, but it's very fine-grained, and it's if you plot it, it's very noisy. So let's see. Wait, these are actually, that's the next file. Okay, so the only thing we do here is actually load the data. We set this up like a pipeline of files that call each other. Here, Dennis did some stuff with time lag, so looking at times before and after, but we didn't actually use that in the final thing. All right. So once we have the data, we have this, we call the other notebook to load all the files. To reduce the noise, we take a time window of, let's see, 30 seconds, and average that. So we go from 5-second data points to 30-second data points, just to reduce the noise. Yeah, so that just and also reduces the number of rows, right? I think we had like 60,000 or 70,000 for a month, so it reduces that a bit.
You collapse it into 30 seconds.
Yeah, exactly.
And RH is what? Relative humidity and atmospheric pressure.
We had it in the previous thing, but
yeah, I was trying to remember.
So, well, it doesn't even say here, right? But it's these things. So this is why it would be nice to have Dennis here, because he knows what all these words means. But yeah, it's just data. And this is sort of what it looks like. So it's a whole bunch of data points. Okay. Then we took inspiration from the notebook 21 in this course. So that's the activity prediction, I think, or is it just
activity detection?
Detection, yeah.
That's Amira Chinese.
Yes. So classifying was the based on the
from the accelerometer readings of the cell phone, whether you're walking, sitting, jumping.
Yeah. So we basically just took that approach. So this is just basically reproducing that notebook, but with our data and for our target. So what we did was, this is a bit complicated. For every row, we look at the past, let's see, 30 minutes of rows. And from that, we compute the mean and standard deviation of all the columns. Okay, but we do that for every row. So this doesn't change the number of rows in our data set. It just adds new columns to it. Okay. So this is just copied from that notebook, computing the mean power, some intermediate stuff to get the standard deviation power.
Do you then remove your first 29 rows?
No, we keep those.
You keep those, but they are
given that we have like 60,000 rows, I think those 29 rows are fine. 60 rows in case.
So the data frame transformers for adding extra columns.
Yeah. No, but also the first rows will just be the average over.
Like the first row will be
nothing.
The average over nothing, and then the second row we just have one.
One
for the average.
Yeah, so those will be a bit weird, but it still works. So now we have this thing. So we have let's call this a lot of columns. Okay, I think in this month there was basically no rain or something. But remember, every row here is 30 seconds, right? But 30 seconds now overlapping in some sense, right? Okay. Yeah, 61,000 rows in this data set. Then we decided to predict 1 hour in the future. Okay. So what we did was for every row, the rows are sorted. We just go, 120 rows down, which would correspond to 1 hour. Look at what the mean power produced there. So 1 hour in the future, what has been the mean power of the last 30 minutes, right? So we're predicting like 32 an hour in the future, kind of. And we're just pulling that value and adding to every row. Okay, so it's the same thing, but with this extra column, future power. use your mean power. Yeah. Again, every row is 30 seconds. Okay, and then we just do random split into 2 parts, the training set and a...
So the future power is your label.
Yes, right, we're doing a regression here, not the labeling. And the notebook 21 is labeling, yeah. But here it's regression. Yeah. But anyway, so we split the dataset 2 parts, 80%, 20%, and yeah, and these now exist. So that when you run it, you will have these variables. I mean, in the next notebook, they will exist. If that makes sense. Right, so in the next notebook, which is now our training notebook, when we run the previous notebook, these train set and test set, they now exist in this notebook. Okay. So vectorizer. So we only use these columns. So the mean power and the standard deviation power. We don't actually use the actual power at that specific instance for our training. Yeah, and then this is just a regression model, I guess, extracted from some notebook from the course. Just doing its thing, right?
It's linear regression.
Yeah, linear regression. I'm not going to go through the code, but basically what went up is wait, okay, let me go. This is actually copied from another notebook, so we go down here. Here's the result after cross-validation, or using cross-validation. So we get the root mean squared error of 5.3 in an R2 of 86. Now, I don't actually know what this means, right, or how good this is in this context, because I don't really know the context. Yeah, it's all right, if I understood it correctly. It means that we are 86% explaining the data or something.
But you can't philosophically say how good it is, just one thing. Right?
Yeah.
One more thing.
Yeah, the other data point is coming. but just, I mean, what does 5? I mean, it corresponds, I mean, this 5, how good that is for standard deviation, depends on what the actual power values are. But they are like between, I think, 0 and 30 or 40 or something. So it might be quite big, I don't know. But yeah, I think Daniel said this was still pretty cool and good results. So I'm trusting him. But then we basically wanted to use gradient boosted trees. So here's now using gradient boosted trees. This again was taken from some notebook of the course, but you know, adapted to what we were doing. Takes 15 minutes to run the train, and we get an R2 of 96 and an RMSE of 2, which according to Dennis is again very, very good. Surprisingly good. So that's cool.
So you don't even have to do deep learning.
We don't even have to do deep learning. I have some more stuff. This is basically the end of the presentation, really. But we've been doing more stuff, and there's more stuff coming below. So I think Mahmoud looked at neural networks a bit, but it didn't really go anywhere and was kind of terrible, like worse than the linear regression. So we just
but also your number of data points is probably too low for any serious deep learning deep network, right?
Is it? Well,
because how many rows do you have in this thing for the
80% of 61,000.
That's
it's not
50,000.
Yeah.
How many variables? How many parameters would we have in the deep learning neural network?
Well, it depends. I don't know.
It's not parameters. It's number of rows, right? Data points.
Yeah, it's a number of data points usually you need. It depends on how deep your network are you doing LSTM. I don't know what, but generally.
And I guess in general, if you did some deep things, then you wouldn't have 15 minutes, then you'd have 15 days.
Yeah, and also, yeah, that's deep. Yeah. So generally, those are very good if you have lots and lots of data.
Yeah.
This gradient boosted regression tree is not good enough for your stuff. That's when I would go further. And also, when there's too much data and too many dimensions, these things break.
Yeah.
These space partition trees break. They don't work well.
So the thing is, here I'm only looking at 1 month. I think it's May this year. I get back to this a bit, but we have more months we could look at. But basically, we decided that there's probably there's a seasonal variation for each month, basically, or a quarter of the year or whatever. So we shouldn't train the same model on different seasons because we don't have to, right? It's better to have one model for every season. Probably. What else? I just want to talk about a bit other things we did. So Dennis spent a lot of time doing autocorrelation. What is this? Yeah, I think it's autocorrelation. So looking at the time series we have and finding how much does it correlate to itself or whatever. And from this you can see like how big time windows can you predict. And
these are the REMA models and stuff you're talking about?
Yeah, I think so. But this is way outside of what anything I understand. And Dennis has a notebook on this as well, but I don't know how to interpret any of it, so I'm not going to present it. So we looked at other things as well, and yeah. And also another thing is that we wanted also to have a different data representation, namely taking, instead of the average over 30 minutes, taking the 30-minute, column of 30 minutes worth of columns, and flattening it to a vector.
Oh,
so to preserve the sort of the time series, because right now we are sort of having this destructive operation of averaging. But digging through the Spark documentation, there is an aggregation function called flatten. It seems to be doing what I want, but I can't actually find any documentation on it. So we didn't get around to try it. So there are other things we wanted to do here, but this worked out. However, out of pure interest, I tried taking our model for May, running it on June, the month after. So here's that. So here's running I think this is the linear regression model on the month after, and you see the results are going down quite a bit, linear regression, and the same story for, here. No wait, it's the other way around, right? This is linear regression right now, and the one above here is the gradient boosted regression tree. So taking it from 1 month and running it on the one after. And you see it's going down quite a bit. And I have the sneaky suspicion that actually taking a random sample from a time series, a uniform random time sample, will just produce the same series, right? Which means
probably just because you have these type of things are probably fairly strongly autocorrelated. So if you know that the wind now, you probably know the wind in 30 seconds.
Not really. So from my understanding, there's not that much correlation going on.
Okay.
Or things can change quite quickly. What I mean is more like if you plot the power level over 1 day, it goes up and then down a bit. Now if you take if you split it into 2 sets randomly, you will have one that goes up and the other set will just have the same pattern to it, right? If you do a uniform random split of a curve, it will just be 2 curves, especially if you are doing this averaging over them or whatever. So I'm thinking we may be sort of overfitting the model a bit because our test set is basically looking like a training set. That's what I'm trying to say. And I think that's why when we move to different months that we see things behaving differently. But it could also be this, seasonal.
You have to train for that month when you hit that month, right? Because you're just doing.
Necessarily.
It depends. I guess if I don't know what.
I think you're right about your. I suspect you're right about your suspicion that you get some overfitting, but from the way you.
I was thinking that the training set looks like the test set, right? Just the curve.
Yeah, because they're from the same month.
No, not only that.
Okay.
They are a time series, right?
Yeah.
But, yeah, exactly. So, because you just sample. You just sample uniform time points.
Yes.
And there's so much dependence. The dependence structure. So it's probably better to cut So I mean, I think what is probably better is to cut large chunks.
Yeah.
And
Maybe splitting on days, doing randomly
Exactly,
taking
yeah.
days over a month as the
Exactly.
training and the test set.
Oh, right. Yeah.
Or even just split it at some.
Yeah.
Just pick 20% contiguous somewhere in the set.
Yeah.
And then train on the rest and use that.
And I think also would be more interesting to actually have data sets over years, multiple years, because then you could use the same. I think you could do the random split just fine, because then you have these different years, not different.
But I think this thing with doing it for different months is not necessarily that important, because I mean the relevant structure that differs from month to month. I mean, you would do this because we have different climates in different months, basically, but you also measure most of these factors, right? You already measure like rainfall and wind. And
yeah.
So I mean, it should be able to basically detect what month it is. The information should already be there.
Yeah,
more or less. So I don't think necessarily it's that important.
I mean, if you train on a full year,
yeah,
right, however, validation choice and stuff, then you can just apply it to the next year.
Yes.
That's what would be more.
Yeah, but we don't know how sensitive the model is to these seasonal changes.
But can you do some clustering beforehand, just to see if you have like great seasons? Because I mean, you could have like 2 seasons or like 1 day.
Yeah, but I mean, in the use case, we know which season it is, so we could just have a library of models and say, oh, it's this month of the year, use this model, because we know which season we're in. Or I mean, we can have it's sunny this month model and then use that average.
But you're
I mean, like Tilo said, since you're already measuring all those things, then hopefully the model would pick up on that, and you wouldn't need to split. Because if you're going, I think one of the points with this is that you can have a somewhat simple model to describe it. And if you start having like one model for each month, and then maybe one model for if this month was sunny or rainy or something like that.
Yeah.
Then you're basically
Kind of
doing
defeats the purpose.
everything twice. You're doing sort of.
I mean, that would. I mean, I guess the best way of trying that is to try to basically classify every data point. Which month is this data point coming from?
Yeah.
I don't know.
I mean, you can do this to detect how well you can detect months.
Yeah, exactly.
Yeah. I think maybe something that might be would have been worth looking at is, rather than doing this averaging, there are various sort of filtering techniques for these type of signals. Yeah, so I mean, you have this high variability when measuring temperature or whatever, and so smoothing these things, there's a lot of classical theory like Kalman filtering and these type of things. So to get rid of this type of noise,
right,
in a way that takes this sort of autocorrelation more into account.
Okay.
So this might actually be faster, both computationally faster, and give you better filters and get rid of some of these other issues.
And there is a 15 HP course on time series alone.
Yeah, 10 HP certainly. I don't remember it. So,
yeah.
So it's a very large topic.
This is the stuff you should telling Dennis basically, because his PhD is in this.
That's good.
Yeah, I think this is it. But I mean, I don't know how good these results are, but Dennis said it was surprisingly good. So I'm happy.
So they can figure out when to turn on the nuclear power plant.
Yeah.
Okay. All right. So there are no other questions. Let's thank Gustav. Okay, our next student presentation is by Yevgen and Dimitri, and they're going to be talking about using Hail for scalable population genomics. Try to sit together so or turn the computer as a talk.
Okay, let's get started. So this is our project. The aim of the project was to create a tutorial for doing genomic data analysis based on this 1000 genomes project. which is described in this Databricks notebook using open source project Hail, which is some like Python package written around some Scala source code. So we wanted to make it in Scala. So this was one reason. The second reason is that this notebook now it's outdated. So some functions which were added later are not exist in this example. Some functions don't work anymore. So most of the time we spend on like digging in the source code because it like poor documented. But yeah, I think we did some success, and we hope that it could be used for some more advanced projects.
Yeah, basically, like originally it's written in Scala, but I think to make it user-friendly, the developers decided to make like a Python wrapper on top. But I think it's probably better for people who know Python than know Scala. But here we wrote everything in Scala. It gives us advantage because we work directly with Scala objects. So the idea is also to develop some function for this Hail project. And now when we work directly with Scala, we're kind of more flexible. And I think it may be useful also for future if somebody wants to understand how it works. It partially overlaps with the presentation I did like a few weeks ago, so I'm not going to stop too much on all these slides, but I just copied them here. So again, it's genomic data is growing very fast. We have a lot of data. It's even predicted that it will be larger than like YouTube and astronomical data. So you can see like it increases a lot. Just like in 2012, we had 1,000 genome, and now we have almost 1 million, I think. Yeah, so it's a lot of data. So in this project, as Evgen mentioned, we used 1,000 genome project, but it's only a subset of that. So we have around 700 samples, I think, and 100,000 variants. So it's quite small, maybe only a few percent of the data. Yeah, and another thing is it becomes cheaper every year, and it becomes affordable. So quite soon, everyone will be able to sequence his or her genome and get all the bad mutation you have and no information about your, like, if you are more prone to some disease. So this is also kind of moving towards personal medicine. So that's why it's important to make another.
Pretty soon dating sites will ask for your genome, I'm sure.
Yeah, so that's why it's important to develop some very efficient tools to analyze this data, and I think, and we also believe that Hail is kind of a very promising project in this regard. So again, this is how the data is generated. So this is a chromosome you can see on top, but of course we cannot get the sequence of the whole chromosome because it's very big. So usually it's chopped into small pieces, and then you can read, the machine can read the nucleotides in every piece, and you get in the end like a pool of these reads. But because there are some mutations which overlap, you can kind of guess how to assemble all these small reads into a larger piece. But in reality, it's even easier because once you assemble the genome, when you have one continuous sequence, you can then just map the reads, new reads. You sequence another genome, so you can take the genome and map to existing genome. So it will be more efficient, and you don't need as much data as you need for assembly. Because here, even if you have 1 or 2 reads, it will still be mapped to the genome because you have like one continuous sequence. And this is how it looks on this picture. You can see all these gray bars. These are just DNA reads, small pieces. It's around 101, I think in this case, yeah, around 100 letters, like nucleotides. So, and this is just a small fragment of genome. So this is the data, it was generated before, we didn't do this. It was aligned. In the end, like based on this alignment, we can get the letters. In each genome, we can see if it's homozygote, so because we have 2 sets of chromosome. So here it's the same letter, let's say A. But here you can see it's heterozygote. So one chromosome has one letter, another chromosome has another letter. And in the end, we can get these letters for many individuals, in this case like 1,000 individuals, and for every position of the genome. And this is stored in so-called variant call format. This is like a big table. Every row is a position of the genome, and every column is a sample. So in HALE, they call it VDS, which is variant dataset, but like it's a transformation of variant call format. And again, you can see this annotation. This is the sample, this is the variant, and in addition to this, they also provide the annotation for variant, annotation for sample, and you can also have kind of global annotation. Yeah, this is what we used. So again, here you can get some introduction about HALE project. Yeah, this is where things started to get complicated, because you can use HALE, which is already available in Databricks, but as Eugen mentioned, it's outdated. So we had to compile our own HALE from the latest source code. So I described it here. It's quite simple. You just need to install the compilers, download the source code, compile and get the jar file. And I also previewed the jar file which I built. We upload it here. So you can also download it. Then you upload it to Databricks. That's a jar file, yeah. And it also works with Spark 2 and Scalable 2.11. So it's, I don't know, but it's also a little bit outdated.
So it doesn't work on Spark 2.2?
No, they have Spark 2.1, I think, also source code, but I think 2.0 is more stable. So, but we tried these different versions, but this one worked, so we just kept this, and this is what they recommend on the website. In addition to this, this HALE project requires different settings for memory, like for parquet, block size, and other things. So you also need to copy this configuration to cluster config. So when you go to, when you create cluster, when you create a cluster, you can specify the additional configuration there. But that was not the end, because after that it worked and we were quite happy until we faced some errors. And those errors, we still don't know why exactly some functions work, others don't. But thanks to Ivan Sadikov, he helped us to figure out what was wrong. Basically, because it was custom jar file which we uploaded, Databricks, like the workers, Spark workers, they didn't see it in some cases. So we had to copy, kind of using the script, copy this jar file to these workers. And the major problem was when you upload the jar file to Databricks, it changes its name. It adds these random numbers at the beginning and at the end. And that was tricky for me because I thought it would have the same name which I used, like when I uploaded, but it didn't, so it didn't work. But using this script, you can figure out the name of the file. Like, because I know that it was HALE or Spark, so you can use this like a string to search for that jar file, and then you can extract the full name, and then you use this full name, and you can copy this jar file to this daemon and like jars, and after that it will work. And this is related to the error here. So if you get this error, this is because you need to copy this jar file. Yeah, so that was the trickiest part, to set up everything on Databricks, but after that, everything worked fine.
Yeah, so then once one has run these two commands, the cluster has to be restarted. And then we can start processing our data. Yeah. Yeah, so we start from creating HALE context. So as I understand, it's just an extension of Spark context. So they just added some additional functionality in order to make it work on Spark. And we set up paths to our data files, annotations, Then we import the data, which is stored on our VCF path, and we use this import VCF function, which is a member of this Hail context object. Then we can do this split multi-allelic variance. I have no idea what it is.
Yeah, like basically most of the functions, they assume that there are only 2 variants per site, but sometimes you can have 3. So, and to make Spark work, or to make Hail work with it, you just split that variant into 2 variants and keep only 2 allele in each. So it's just something.
The most frequent one.
Yeah, usually most of the sites, they are like biallelic. There are only mostly 2 letters, but in some cases you may have 3, and they want to keep it. So you can also apply this function because you will get error later in some analysis.
And as I understand, in this case, it's already split. Right?
Yeah, this is the data we use here. It's already available in Databricks. It's human genome data, and it was already splitted. But if you have your own data, you will need to apply this function.
Then we import annotations. We can print it. First, we convert it to data frame. Let's use display function, built-in function from Databricks. So this is sample, number of sample. This comes from these populations, Great Britain.
Yeah.
Like superpopulation is Europe. Then if it's female or male.
Yeah,
Hair color.
broken hair. What's this? I don't know. That's a phenotype, but why it's purple?
Caffeine consumption.
Yeah. Yeah, some phenotypic data. Like you can use any data there. You can put eye color or whatever you hate.
Yeah, then we apply annotation schema to our VDS. We can print it. So if we use just print, then it has such an output. We can make it more readable. There is a method for this schema ready JSON. Then we can read it more in more details. And we can print global schema, which is empty.
Yeah, you cannot annotate it. No problem.
There's this VDS object, which is variant dataset. It has some fields. We can just press dot tab, and we can see what is provided. For example, we can print out number of samples. In this case, we have 710. Then we can plot histogram of population frequencies, for example. And we see that all this population has like the same number of samples.
This should count 1,000. 1,000.
No, I think it's absolute number. It's on average 25 samples.
No, I know, but
it's 110.
Is it 710 total?
Yeah,
number
it's subsample from 1,000 project, but not all samples. I think that they throw away the bad samples.
Yeah, then we can do some summary of what we have in this data set. So it prints it like this.
Yeah.
As I understand, summary result is some built-in structure package.
Yeah, so I can click over here. So when you just print it like this, it's not that pretty, but in this order you can read this information. So again, it's number of samples, number of variants. Then you can also see core rate. So these are the different chromosomes. not really. So you can access all of them with this summarize function, but if you want, you can also get the specific function. So you just save this summarized object, and then again, if you put this object, you type dot, and then you press the tab, you can extract specific function. You can just see the correlate, or you can see the number of samples as we've done before. So I'm not going to stop too much on that. The core part of this project was to filter this data, and we filtered like genotypes. So the genotype is a specific cell in this data set, variant is a row, and sample is a specific column. Yeah, and there are many annotation scores for all these variants. So they usually encoded in different parameters. One of them is Preprocessing quality score, which is like genomic specific annotation score. You can read more in this Wikipedia page if you want. But basically what we do, we extract this, in this case, the genotype annotation in this big table. So you can see it's, yeah, it is quite large. And in this case, it's also a bit of a problem because basically what it does, it takes each row and concatenate it into a very, very large table. But we wanted just to define the cutoffs for this. So we use some SQL here. So we extracted the depth. It's how many reads we have for each genotype, and also genotype quality. And genotype quality is this Preprocessing score. So we obtained this table, but because it's very big, and here we're only interested in the distribution to define the cutoff, so to see where we can cut, but at the same time to keep the major part of the data. So we limited to 3,000 rows, and we also randomly sampled them using this random SQL function here. So basically, we just sampled every 10th, no, every 100 rows of this big table, and then we just use Databricks display function. So if you click on plot, so here you can select what you want. So we put the genotype quality, but you can also use depth. If you drag and drop here depth, then you can see the distribution for depth. Yeah, and from here you can see that most of the genotypes, they covered like 15. Oh, genotype quality, sorry, yeah. This is first like human genome 1000 project, so the genotype quality is not very good here. According to the current day standards, it should be more than 30. But you can see here it was quite bad if you compare it to today's data. But you can still see that we can maybe cut like 4 or 5. So we can cut this part, which is not very good. But most of the data is more or less good. So here we defined, when we look at this distribution, we kind of decided on the cutoffs. So we decided to cut if the genotype is covered by more than 2 reads, and the genotype quality is more than 10. Yeah, actually here I cut it more. So all this was thrown away. So we filtered this, then we can check the correlate again. We use again summarize function, but we don't call all the summary statistics, we just call correlate. And here you can see it decreased a little bit. Previously it was like 84%, but now we have 84. 98 it was before, now it's 84. Yeah, next we filtered samples. So in previous step we just filtered specific cells. Now we can look if some sample have most of the cells removed, for example. So maybe it makes sense to throw away that sample. We annotated the samples because we changed the genotypes in the variant dataset, so we need to generate new annotation to count all the cells which we removed, for example, if it was missing data. So we've done that. Then again, we extracted this annotation to the table. So you can see it here. And here we have different statistics for each sample, including the correlate for every sample. So we have seen the overall correlate before, but we can also obtain it for every sample. And again, we use display function to visualize different statistics. So here you can see that most of the samples are called is more than 90%. I don't know why it uses this annotation in Databricks, but it's 0.9 basically. But using this shortcut or something. So from this distribution, we can see, okay, we have some samples which are not as good as other samples, so we can throw away, for example, this tail. And we can do it for various statistics. But here, I decided to keep like all the samples which have more than 60% correlate success. So if sample has like more than 40% of missing data, we will not use it, right? Or more than 60. Yeah, and after that, we had 710 samples, and now we have 667 samples. Yeah, and here it's just to verify that our filter worked. So we take filter data set, extract the annotation scores, and check the distribution, and you can see that we don't have samples with correlate less than 60%.
600 milli-units.
Okay, yeah. Yeah, and the last step is to filter variants. So we now remove specific cells, we filter it for samples, and now we can also filter it for rows for bad variants. Again, we annotate the variant, and we also use cache function to speed up a little bit because now it's loaded into memory. Basically, the procedure is the same. We generate the table for these annotation scores. Then we build distribution for different statistics. And based on this distribution and also some recommendation, I put the link at the beginning. You can read about all these annotation statistics. There are some recommended cutoffs, but because the data quality can vary a lot, so you need to adjust it a little bit to find trade-off between the amount you throw away and amount you keep. So in this data, we decided on these cutoffs, we filtered it. Yeah, and in the end, we are left well, still like around 10,000 variants.
So these are equivalent to SNPs, are they?
Yes, in this data set, every variant is a SNP, but you can also have non-variable sites. But in this case, they're all variable. So we remove not that many, like whatever it's 400, 300 sites with this filtering criteria. Yeah, and again, we checked if our filter worked, because sometimes if you put the sign in the wrong direction or something, you can remove a lot. So it's better to always verify your filtering criteria and make sure that it worked as you wanted. And after that, you can write this VDS to a file. So it's a filtered data set, and you can use it for whatever analysis you want in the future. So yeah, we wrote it in this VDS file, and then we read it back to our VDS filtered data set. We have not done much analysis here. We just performed a simple PCA analysis because it will be easier for most of you to understand. But I think here again we'll explain you.
Yeah, so we are taking our filtered variant data set. We apply PCA, and we chose two components just in order to visualize it. Then so the result of this PCA, yes.
So what representation are you applying the PCA on? So what?
It's a sample.
So what's the vector? What's the single observation now?
It's every sample for every sample.
So every sample means it's an individual genome.
Yeah.
And
Okay, and you look at the site at the sites where you have variants. And
yeah.
For every site, you have one component in the vector. Or...
No, you have a component for every sample. No.
As I understand, it takes just like from this schema annotation.
Yeah, we have like after filtering 600 something samples, so it will be around 600 components.
Oh, okay. So you want to look at the sites. You do PCA on the sites. Or so what's one individual? Yeah, what's one point?
So the row is the individual.
The column is in
The column is the individual.
Yeah.
And every row is a
variant.
variant.
Yeah, okay, yeah.
Call it a
Yeah,
The
so you're
row-
looking
the
at the
row
set
is
of
a-
variables.
is a
Yeah.
feature vector.
Okay, yeah.
Yeah, so it produced so this object VDS from the Scala source code, so it has a PCA method itself, and it produces variant dataset as an output, but just with some additional component which is called PCA. So it's not like data frame as an output, it's variant dataset again with just PCA added.
It's probably using a transformer.
So in order to make it as a data frame, we apply this built-in functionality. And it gives us a data set like this, but by default, all these column names, they are separated with dots. And when we did an SQL, it didn't understand. So before extracting this PCA component into a data frame, we had to rename the columns. So with this command, we replace dots with underscore. And yeah, so this is data frame like this. And then we do SQL. So we extract population and we extract component 1 and component 2. It looks like this. And then we did a visualization. So here we see that we have like clusters of different populations. And also we experienced an issue with these display functions in this case. So first, when we apply display, it produces a table. And then when we wanted to have a graph representation, so when we chose the scatter plot, it crashed.
Oh.
Yeah. So in order to make this plot, we chose a line plot first, and then we changed it to the scatter.
There is a bug.
So
I think it's some bug in database.
Yeah.
Because we tried it on different computers and other servers. Yeah, so this is actually analysis, but also we can use R.
I guess you can hit that send feedback button and
Yeah, when we had error, we clicked on.
Yeah.
give them.
So it's allowed to make an SQL query and to put the results into R data frame. And then if we use ggplot2 package, we can produce such a plot.
Yeah, another
What's
advantage
the
of
purple one?
The purple is so
I think the first two it's Africa, different part of Africa. Then we have East Asia, Europe, and probably South Africa. I don't know.
South American.
Maybe.
I mean, the American is the one that's spread out from everywhere because Americans are from everywhere, right?
Can you go to the Databricks?
Yeah, the
built-in
Databricks
and
display is a little better for just seeing the.
Yeah, I don't remember exactly this annotation, but I think those
I
two
think
just
it's-
separate.
yeah,
I
it's
think
there.
these are
So
two
Europe,
African probably.
East Asia,
Maybe hit
America.
America is spread out.
which makes sense. Yeah, SAS.
South Asia?
South Asia, maybe, yeah.
Yeah, possible.
So you guys didn't read all the details of the data set. No, this.
Yeah, I don't remember exactly this annotation, but just to see that we are not homogeneous.
Yeah.
There is strong population structure. There is actually another study when they look at European populations, and if you do PCA, you almost get like European map by countries. So
Yeah.
it shows that humans don't move much.
I mean, the 23andMe, I believe, like there was a Science for Nature paper a couple months ago, right? They did this crazy thing because they have the genomes of all these people who want their ancestry, and they mapped it down all over the US map. and it's bang on like towns in Pennsylvania, everything like super micro because it's the full genome based thing. Yeah, so that's makes sense, right? If you only breed with your neighbors for 3 or 4 generations, that's basically it.
Well, we started to move just recently, so yeah, it's not like many people move. Just because we're in academia, it seems like we travel a lot, but most people don't move. Yeah, so this is just a simple simple workflow, and it's just the beginning. There are not that many functions in Hail to actually do a lot of analysis. Most of them is just to filter and do some simple things. And to generate the data, like these mapping steps and alignment add-on, I think is a little bit better. Like if you want to do this distributed computing, of course there are some other packages, but Adam is more focused on this part, like to actually generate this VCF file. And I think Hail is kind of better to actually work with variant format. And here I presented just in summary how you filter, but it's up to you what's your priority. You can filter it in a little bit different order, but I think this is the most logical. You first remove bad genotypes, so you remove specific cells, then you remove bad samples. But usually, like at least in my case, I never remove samples because I don't have that many samples, just because it's human genome, so you can afford to throw away some samples, but usually just remove variants and some genotypes. Yeah, and it was a little bit of pain to make it work on Databricks. Maybe it's because we are not very experienced and we don't see exactly what happens, but even reading the log files, it was not very straightforward, because Ivan sent, like, you basically need to write a short script to extract and read the log files from Databricks to find what was the problem.
Yeah, but Ivan has been working on this stuff for a year and a half or something every day. He's like a hardcore data engineer.
But you can, of course, install Hail locally on computer, and it uses Jupyter Notebook. But then you can work with PySpark. You need PySpark, and then you can do everything with it.
So from here to, you already have what I guess mathematical pulpgen people will call a binary incidence matrix, which is like a slightly different, where every row is the individual, okay, and every column is a SNP.
Yeah,
it's basically here, right?
But you need to transpose.
Yeah.
Like VDS, it contains all the information.
Yeah.
Even like when we run PCA, it adds information there.
Yeah.
So they try to keep everything. So if you find something interesting, you can go back and check everything. Even this filter criteria, here we removed samples, so it would be missing data, but you can also just apply filter. So you add extra value to each genotype, which says that it's low quality, for example. And then like you can keep, if you want, the genotype, and you can go back and remove filter.
So for example, from here, by reading like some of the chapters in Wakely's book, you should be able to get from the binary incidence matrix to the total number of segregating sites, which is just the number of columns.
Yeah, that you can calculate.
Heterozygosity, heterogeneity, FSD.
With
FSD.
GMSD, FSD, it will be, I'm not sure it's implemented yet, but simple things like heterozygosity, you can get.
Well, if you implement those, then that's basically material for the genomics course.
Yeah, I think they have like Hardy-Weinberg equilibrium, some very simple things to calculate, and they mostly focus on genome-wide association studies. That's what most people do. I know it's very fancy. Like basically the idea, you try to find association between phenotype and some genotype variants. So as you have seen, there is this caffeine consumption, like a phenotype. So in the tutorial, they also tried to do this association. I personally am still very skeptical about all this. I think it's still in infancy. It's very difficult to pick up everything, because in human genetics we have these quantitative traits. So it's
complex as well.
Yeah, it's variation of many variables, and if you change one variant here, it completely affects the effect of other variants. So it's tricky, but it's a future for data science to figure out what's going on. yeah, when the first like genome was sequenced, everyone, oh, now we know everything, we can understand, we can treat all diseases, we can predict if you are like you'll get sick or something, but it didn't happen. Yeah.
So I'm just a little confused again, but so when you do the PCA projection, you do it on variants. Yeah.
No.
And
So the number of dots in the PCA plot, is that equal to the sample size?
Yes.
Yeah.
Okay,
So they're transposing it again.
I don't know.
I mean, so the PCA is taking the variant size and reducing the, I don't know how many variants that you have to
okay.
choose on.
Oh, I thought it was
So
the other way around,
Yeah,
but then I was confused about the countries.
No, I
Okay,
know you
then-
misunderstood
okay,
it, but
yeah. Okay,
I
good.
mean, the
No,
PCA
here,
you are
like-
calling is a Hale function, basically.
Yeah, so every dot is a sample, and then we just cover it
Yeah.
by this annotation.
I'm pretty sure Hale is calling the MLlibs PCA implementation. Right?
I don't know if we haven't
Otherwise it won't scale.
checked.
It's a bit strange to do PCA though, because it's basically a binary vector, right?
Yeah, most of the sites. Yeah.
Because I mean, PCA relies sort of on a vector space structure rather than just. I mean, it works, but it seems more natural to do some sort of clustering with the Hamming distance. But maybe this would be basically equivalent.
Actually, we wanted to do more analysis, but as I
Yeah.
said, most of the time it was just
Figuring out the jar.
Yeah.
No, but I mean, this can continue.
And also to understand what was written, because so the workflow was we opened a Python file which was just so function we just wrapper it around with Scala, and we go through this Python code. Okay, this Scala functions. Then we went to Scala code, but at some point all the Python code disappeared from this GitHub.
Oh, they updated it.
So,
Yeah.
so they updated is like so many functions.
So these are the Broad Institute guys from Boston. Who's behind Hale project?
I think it's together between MIT and Harvard.
Yeah,
Medical
that's the
School.
Broad Institute.
Yeah.
So I now have a question for you again. What's the worst? Trying to get Scala stuff running on Databricks, or trying to get R stuff running on OS X?
Yeah.
No, for us it was difficult because there is a little bit of documentation for Python, but there is no Scala documentation, and even the code is like it's annotated, but not very clear.
Scala code is not commented.
Well, yeah.
It
maybe
is in
it's-
some parts.
Yeah, but most functions are not commented.
Yeah, they're probably changing all of it live, right? But it's actively being developed, right?
Yeah, they work because I don't know. I don't think it's outdated now because they have not changed the version of the program.
Yeah.
But Evgen says that he has seen that they updated.
So at least they restructured all the Python side.
Okay.
But it happened in the middle of our project, so we still we use the previous code. But I don't think. I think maybe they just move it into different folder, but it should not change much.
Good to write down the exact Git. What's the
Version number.
yes, but what's it called? The hash.
Yeah.
The
You have a hash.
exactly, but it's called the revision.
Yeah.
So it's good to know the exact revision and then stay with the one revision. So that you don't-
But once we produce this variant dataset file with Scala code, and we can transform it into data frame, then
Yeah.
we can call all this.
Yeah, exactly. That's where I would just stop there and just go into Spark.
Yeah, and here it's also in original tutorials they used Python pandas to do all these plots and all these scores, but here we had to
Yeah, we
use
can
data
use
frames
this
and
Spark
SQL.
ML read.
Yeah. Are there any
We
other
hope
questions?
it will be useful.
Yeah, I think it'll be useful even for the course I'm planning to do, maybe with Dimitri in spring. Definitely save me lots and lots of time.
Oh, it was useful for us. This,
But just to see that I understand. Your idea was you took this failed project and then you wanted to use that, but without using their Python wrappers.
Yes.
Yeah.
To go to their implementation things in Scala and just call that directly.
Yeah.
But you still needed to know what the functions were called.
Yes.
And stuff like that. Yeah.
So they kind of reverse engineered the comments from the Python wrap comments.
Yeah. It's sort of.
Are there any other questions for Dmitry? Okay, if not, let's thank them again. So we're all done for the course. So please fill out the survey if you haven't. I think it's from SurveyMonkey. I sent from my Gmail.
And we need actually you should also remind, because it ended up in spam folder for me.
Which one?
The survey you sent.
Oh, it went to spam folder.
Yeah, I checked
Oh,
it,
right.
so I wanted to tell you and I forgot.
Okay,
But, I don't know why, maybe because it's from Gmail or maybe.
Yeah, it's SurveyMonkey, right?
Yeah.
I would, in general, put